Welcome to the Turtle Time Activity Training video. This instructional video will provide background for Turtle Time, an activity demonstration, and some tips for engaging audiences. With Turtle Time, participants will be learning perhaps the most important word in evolutionary biology, homology. Homology is simply a characteristic or trait that is shared among organisms because the trait was inherited from a common ancestor. For example, the forelimbs of humans, dogs, birds, and whales are homologous because they all descended from a common ancestor long ago. Scientists can use homology to hypothesize about how different species are related to one another. In this phylogenetic tree, we can see how shared traits, or synapomorphies, can be used to group distantly related species. Mammals all share fur and mammary glands. That is why mouse and chimpanzee are grouped. Fur and mammary glands are synapomorphies that unite mammals. Similarly, the presence of a gizzard unites crocodiles and pigeons. The participant will be figuring out how 8 different species of sea turtles are related to each other using homology. This is done using a dichotomous key, which asks a yes or no question about the presence of a particular trait. Let's start from the top. The first question asks, are there plates on the top of the shell? The participant will divide the turtles up by whether plates are present or not. 7 of the turtles have plates, and 1 does not, the leatherback. The presence of plates is a synapomorphy for the 7 other turtles. We can use this information to start building the tree. The trait of having plates unites all of the turtles except the leatherback. We can map the trait of plate presence or absence to the branches and place the leatherback outside of the grouping of the other turtles. We've just made our first homology assignment. The next question the key asks is, how many middle plates are there? The options are 5 or more than 5 plates. Only the olive ridley turtle has more than 5 plates, with 5 plates being a synapomorphy that unites the other 6 turtles. We can then map the traits and place the olive ridley turtle in its appropriate location on the tree. When the phylogeny is complete, you can tell the participant that they have generated a hypothesis for how each of the turtles are related to one another. It's important to relay that every phylogenetic tree is a hypothesis, and the relationships of species have the potential to change if new information is used, like DNA sequencing data. Depending on the age of the participant, you may want to focus on different aspects of the activity. Phylogenetic systematics may be overwhelming for a younger audience, so in those cases you should emphasize using their detective skills to interpret a dichotomous key and how all living organisms are related to one another, not just turtles. For an older audience, you may want to have a more in-depth discussion about form and function and how systematics is a powerful tool to test a hypothesis of relatedness. Good luck with the activity.